Another confounding variable in this whole story: what endpoints should we be looking at? What are endocrine disrupting compounds affecting within the body? We started off with the focus being on estrogens because of things like intersex fish and DTT metabolites with thought to be some estrogenic activity affecting sexual development in alligators. Bisphenol A leaking out of cell plastic, which are affecting cell cultures. Declining sperm counts, could they be explained by estrogens? But all of these effects, intersex fish, abnormal sexual development in alligators, the cell culture effects, declining sperm counts, are all easily monitored. They're obvious. What about less obvious ones? More subtle effects within the body. They may still be very significant, but we just don't notice them. Metabolic effects, behavioural effects, maybe a whole spectrum of responses. We could choose endless examples here, but here's a nice one. Bisphenol A exposure and behaviour. This is in zebrafish, so we're going to work up through zebrafish, then mice, and then look at humans. And this study showed that bisphenol A exposure induces behavioural changes in larval zebrafish. So this is looking during development, 0 hours post-fertilisation, 10 hours, 16 hours, 24 hours, 36 hours. This is the period of neurogenesis within the hypothalamus, and they were looking at activity, and they looked at activity traces. These are the control fish or control larval fish, which are not doing very much at all, but as you increase the exposure levels, or the time of exposure, here this early exposure, 10 to 16 hours post-fertilisation, a lot of activity, but much more if you expose to bisphenol A bit later, or later still. So there's no doubt that bisphenol A exposure induces changes in behavioural effects in larval zebrafish. And we won't go into the detail of this experiment, but this was showed that bisphenol A exposure alters behaviour and brain development in mice. So not just zebrafish, mice as well. And look, the effects occurred after rodents consumed the chemical. Consumed, so they're taking it in orally in levels 10 to 20-fold below the recommended daily dose for humans. So it looks as though the developing brain may be quite sensitive to bisphenol A. And on to humans. This isn't bisphenol A this time, this happens to be phthalates. But the general principle applies. The prenatal and childhood exposures to things like phthalates may well affect brain development. And in this case, prenatal exposure to certain phthalates was associated with lower motor scores, measured at age 11 in girls, and postnatal exposure in boys was seemed to affect motor proficiency. And it would be wrong to end this section without mentioning that endocrine disrupting compounds might subtly affect things like metabolism. They could act as obesogens, generating obesity. How? Well, perhaps by affecting fat development, by altering metabolism among metabolic set points, and by altering the appetite and food intake. Let's look at another issue which confounds our easy interpretation of the effects of endocrine disrupting compounds on the body. Looking at single compounds is perhaps quite easy. We can identify the biological actions of the compounds on cells, the mechanisms through which they work, and maybe identify the exposure levels generally. But why concentrate on the effects of single compounds when in reality we're all exposed to many compounds in different amounts all at the same time? So mixtures are more important perhaps. In terms of real effects, biological effects, understanding the effects of mixtures is much more important than the effects of single compounds all on their own. The fact that we are all exposed to different compounds is well illustrated by this study, who looked at 52 different chemicals in the blood of pregnant women in the United States. They looked at 54 women and a number of different types of chemicals, heavy metals, pesticides, phthalates, and each lady had a mixture of these compounds, but in various amounts. So the question is quite simple. If we're exposed to mixtures of compounds, what is the sum total of their effect? It seems a simple question, but trying to answer it is much more difficult. To do it experimentally, you have to choose particular compounds. Which do you choose? For each compound, which dose or range of doses do you use? Which range of doses for each compound do you use in combination with any other compound? Experimentally quite difficult, in fact a huge task. This is an example of some of the complexity that can arise. This was a project funded by the European Union looking at the effects of mixtures of endocrine disrupting compounds on various endpoints if you administer them to pregnant animals, pregnant rats. They had three main experimental groups: a mixture of 4 estrogenic compounds, a mixture of 8 anti-androgenic compounds, and a complete mixture of some estrogens, some anti-androgens, and paracetamol as well. And all these mixtures were given by oral gavage to rats throughout their gestation and lactation. Oral gavage, that actually allows you to give a set amount orally into each animal. And the results were clear. The offspring showed a whole variety of effects characteristic of interference with normal male reproductive development. Reduced male anogenital distance, increased nipple retention. Normally in male rodents they lose nipples. Decreased rates of the prostate, changes in the mammary development and changes in the prostate, reduced sperm counts in adults, all characteristic of the sort of testicular dysgenesis syndrome which occurs in humans. So the take-home message is clear. We know that everyone is exposed to different synthetic chemicals. These mixtures will be in different combinations and in different amounts and in different times of their lives in different parts of the world, but we're all exposed. And it's easy to see that different mixtures might have different effects. If each chemical is working by one or more different pathways, then these pathways may overlap with each other, interfere with each other, enhance each other. Here's a hypothetical suggestion about how a mixture of bisphenols and phthalates might interfere with various processes within the body. They might be exerting weak estrogenic activity through the estrogen receptors. At the same time, they may be exerting anti-androgenic activity by blocking androgen, testosterone and dihydrotestosterone action at the endogenous androgen receptor. At the same time, they may be affecting various aspects of metabolism, like lipid accumulation within cells, through insulin resistance, the release of things like leptin. How? By working via PPARs and RXR. They may also be working via the reactive oxygen and nitrogen pathways, affecting inflammation and insulin resistance, all of which combine together to affect the various developmental pathways which these things might be destined to undertake. The next question: does it matter when we have exposure to the endocrine disrupting compounds? Are sometimes more critical than others? We have already seen that the bioactivity of individual endocrine disrupting compounds is generally much weaker than any of the endogenous signals. But even if that's the case, effects would still be expected if there was exposure to the compounds in very large amounts, so this would overcome their weak activity, over a long period of times when any small effects could be cumulative, and or at critical periods of development when key decision-making processes were taking place. The fertilized egg is totipotent. It can produce any cell in the body, but gradually differentiation takes place into the inner cell mass and the trophectoderm, and the embryonic stem cells then form the embryo, and then they start differentiating into each of the various cell types. The trophoblast differentiates into the placenta. Here we see the development of Xenopus, the South African clawed toad. At various developmental stages, decisions are made along cell lines where they split and split again and split again and split again. So the fertilized egg makes decisions And these are irreversible pathways. Some cells go towards the neoectoderm, and at later stages they make other decisions so that the cell type now gets narrower and narrower and narrower. Neoectoderm, non-neural ectoderm, mesoderm, endoderm at different developmental stages. And here we can see how the cell type specific gene activity changes over time. We're looking, the blue colours are what is at the early stages of development, and all the cells, the gene activity is very similar in all the different cell lines, but as we go later and later in development, the different cell lines and the different cell types are showing different and their own specific gene activity patterns. Anything which affects these decision-making processes, which way the cells are going to go, is going to have long-term effects in developmental profiles. Let's look at an example relevant to male reproductive health. Sertoli cells. This is a diagrammatic picture of a Sertoli cell and the spermatogonia that it supports. The Sertoli cells line the seminiferous tubules. This is the inside of the tubule where the sperm are released, and this is the outside of the tubule where the blood vessels are and the Leydig cells are. The blue, this is one Sertoli cell, here's another Sertoli cell next door to it, here's another Sertoli cell next door to it. As the spermatogonia divide and develop towards sperm, they migrate up between adjacent Sertoli cells. Eventually, the spermatozoa are released into the lumen of the seminiferous tubule and then go down towards the rete testis and the epididymis. So this is one Sertoli cell supporting developing sperm. Now the argument goes that the number of Sertoli cells in the testis determines sperm production. Why? Because each Sertoli cell can only support a limited number of developing sperm at any one time. So the more Sertoli cells you have, the more sperm you can actually produce. If Sertoli cell number is important in adulthood in determining how many sperm you produce, then where do the Sertoli cells come from? Well, we can follow them back into fetal life. They proliferate in fetal life and early neonatal life, then the proliferation more or less stops. There's another period of proliferation and a final terminal differentiation as they go through puberty into adulthood, and from then on they just support spermatogenic function. So anything which affects Sertoli cell number here, this early phase, is going to have major effects up here in adulthood and sperm production. So what we've said so far is the number of Sertoli cells in the testis is fixed by puberty, determined by what happens early on in life. The number of Sertoli cells in adulthood determines the number of sperm that the adult will produce, and that will affect his fertility. So one site of action of endocrine disrupting chemicals, in the testis anyway, could be on the early proliferation of these Sertoli cells. But as well as that, one could easily envisage that they can affect Leydig cells. Leydig cell proliferation in early life, if you affect that and their ability to produce testosterone, or the ability of the reproductive tract to respond to the testosterone, that will affect male reproductive tract development. So simply combining these two pathways, the potential action of endocrine disrupting chemicals on Sertoli cell number and the potential action on Leydig cells and the ability to respond to testosterone, can explain the testicular dystenosis syndrome. It can explain fertility, and it can explain effects on the reproductive tract, such as the cryptorchidism, undescended testes, hyperspadias. Both of these are developmental problems, semen parameters, by the effects on Sertoli cells, or testicular germ cell cancer, which is often believed to originate by early life effects. Here we're looking at the time course of events in the male fetus as it develops. This is the time course in humans in terms of weeks, 8, 10, 12, 14, 16, 18, 20, and in rats this is in days, 15, 16, 17, 18, 19 days of development. The same events occur in humans as in rats. The big blue area represents the testicular testosterone secretion, which you can see peaks about 16 or 18 weeks in the human, and towards the end of pregnancy, birth is in 21 days in a rat. So this is testosterone. What else is happening? Well, all kinds of things. The Wolffian duct is present and it starts differentiating here. The prostate formation, the penis differentiation, the urethral development, anogenital distance, descent of the testes. So when is this whole system sensitive to perturbation? If you use anti-androgens, you can investigate it. You can inject a rat, for instance, with anti-androgens on day 15 or day 16 or for a period of time here or for a period of time here, and you can see what effects giving anti-androgens can have. Such experiments demonstrated very clearly that anti-androgens weren't effective if given before day 15 of a rat or after about day 20. During that window in the middle, though, the whole system was sensitive to perturbation, affecting testicular descent, the internal male sex ducts, prostate development, penis differentiation, anogenital distance. This was a sensitive period. It was called the masculinization programming window, between a day about 16 to 19 in the rat, which would correspond in the human to about weeks 10 to 18. And exposure during this period to endocrine disrupting compounds like dibutyl phthalate also induces male reproductive tract problems later on in life. Let's look at some real results. These are looking at dibutyl phthalate exposed testes at 21.5 days of development. So this is actually at time of birth, and you can see the red. Each red represents one Sertoli cell. And you can see that they're organised as though they're surrounding a seminiferous tubule. The Leydig cells are between. Those are the control. That's what it should look like. These are rats which have been exposed during days 15 to 18 to dibutyl phthalate, and you can see the whole thing is much more disorganised. The Sertoli cells are now scattered, not forming nice tubules, and the Leydig cells are all over the place too, but the Sertoli cells are no longer in tubules. If you leave the exposure to dibutyl phthalate later, then there's not a problem. Here's the Sertoli cells in nice tubules again, with the Leydig cells between them. And the results can be seen in the adult too. This is a cross-section of the testis of an adult having been treated with dibutyl phthalate during the masculinization programming window. This is what a normal tubule should look like, and they've got some. So the Sertoli cells are on the outside and developing sperm on the inside, but there are also a large number of these Sertoli cell only tubules. No sperm, no sperm, no sperm. This is actually a picture of Sertoli cell only tubules in a human testis. Adult men with testicular dysgenesis disorders often exhibit such tubules. Here they are, Sertoli cell only, no sperm in the middle, no sperm in the middle, no sperm in the middle. Here's a normal tubule with lots of developing sperm. Another one, lots of developing sperm. So the take-home message of this section is that endocrine disrupting chemicals are particularly likely to have effects early on in development, when cells are making critical decisions about which way they're going to go, how they're going to differentiate. Any small perturbations at this time will affect long-term developmental pathways and adult characteristics. So early actions of, for instance, in the testis, endocrine disrupting chemicals on Sertoli cells or testosterone action or Leydig cells can easily give rise to long-term developmental effects in the adult. Now this is a very important question. Can endocrine disrupting compounds have effects not just on one generation, but in their offspring of that generation, and the offspring, and the offspring, and the offspring further down the line? It is easy to envisage that the compounds can affect the adults. It is easy to envisage that the compounds can affect the fetus. It is easy to envisage that the compounds could affect the germ cells within that developing fetus and therefore have effects in the grandchildren. But what about further on down the line? The children of those grandchildren. They're not directly exposed to any of the endocrine disrupting compounds. None of their cells have ever seen them. Could they still be having an effect? If they are having an effect, how could such effects be mediated? Here we come into epigenetics. The story about the transgenerational effects of endocrine disrupting compounds really started in 2005. They were looking at whether the exposure of a pregnant rat to a fungicide or a pesticide, which had a variety of effects on steroid receptors, estrogenic, anti-estrogenic, anti-androgenic, this one was anti-androgenic too, whether exposure to such compounds would have effects on not only her offspring, but when they grew up, on the offspring of her, and when they grew up, the offspring, and when they grew up, and down through the generations. And the surprising thing was, effects were still seen in the 4th generation. This was amazing. They didn't really expect that a single exposure of the pregnant female back up here would still be having effects generation after generation after generation. None of these had seen the chemicals directly. And what could be the explanation? Epigenetic modifications. And there was evidence there was epigenetic modifications of the sperm DNA which was being passed down generation after generation after generation after generation from that single exposure to these endocrine disrupting compounds. The significance of epigenetics is now known to be huge throughout the whole of biology. Epigenetic inheritance is the transmission of epigenetic marks from the parent to the offspring without any alteration of the primary structure of the DNA. Now, such epigenetic markers can include modifications, e.g. acetylation, of the histone tail. The acetylation can loosen the DNA coil, exposing hidden genes so they can then become active. You can also have alteration of the methyl groups, addition or deletion of methyl groups. Methyl groups act like a roadblock, stopping the enzymes from reading the DNA. There's also the possibility that microRNAs are also involved. MicroRNAs are non-coding RNAs which can interfere with normal messenger RNA and prevent specific proteins being produced. While initial studies concentrated on germline inheritance, whereby epigenetic changes were passed down in the sperm or the eggs, down through the generations, this doesn't have to be the case. It became obvious that many environmental factors could induce transgenerational changes. Nutrition, obesity, stress during pregnancy, of course the exposure to endocrine disrupting compounds. Some of these may be causing epigenetic effects down through the germline, but in others the effects were actually due to changing in, for instance, the mother's metabolism, which would affect the development of the offspring and the physiology of the offspring when they grew up with their altered epigenetic markers. They would then have effects on the development and physiology of their own offspring, and that in turn would be passed down to the next generation, which would be affecting the development and physiology of their offspring. So passing down not through the germline but through epigenetic dependent changes in somatic cells. And here's another typical example. Here mice were exposed to tributyl tin, which is an endocrine disrupting compound, in their drinking water during pregnancy and while lactating. And what happened? The effects were seen down to the F4 at least. What did they see? Tributyl tin exposure just up here induced global changes in DNA methylation and altered expression of metabolism relevant genes. And the descendants, even down here, were leptin resistant, predisposed to obesity when dietary fat is increased. Is there any evidence that transgenerational effects also occurs in humans? Well, the trouble is with humans, their generation time is much slower than in rodents, so the evidence is very slow to accumulate. But it is now beginning to accumulate in some circumstances. Here are women who are exposed to diethylstilbestrol during their pregnancy in the second part of last century. So a number of families, the women took DES for one or more of their pregnancies. Here they're exposed pregnancies. This was the non-exposed pregnancies. And if you look at the pregnancies in which problems occurred, then it was only in the DES exposed pregnancies that, if you look at the children, some of the children had hyperspadias in the males, and the daughters had problems with the development of their normal reproductive tract. Going down to the next generation, it was the grandsons of these original mothers who showed hyperspadias, at least some of them did. None of these others did. So exposure up here, one generation, 2 generations down, there were still developmental problems. So the take-home message of this section is that endocrine disrupting compounds may not just affect the adult body, but they may be passed down from generation to generation through epigenetic mechanisms. And with each generation, they can be subject to new exposures as well, which may be having other effects. Another confounding issue. We are not alone. We have within us trillions of other cells, not belonging to us directly, but belonging to the microbiome, all over the body. How do they handle any of the compounds we're exposed to? Do they modify endocrine disrupting compounds? Do they detoxify them? Or can they convert them to something more active? Here's a good example. Genistein and Dedzine are both phytoestrogens coming from soy. Dedzine can be converted by our gut bacteria to equol. And equol isn't too dissimilar at all to estradiol, and equol can have significant estrogenic effects. One of the themes running throughout our look at endocrine disrupting chemicals has been their ability possibly to damage fertility and male reproductive health. The complication here is that a number of other things can as well. Alcohol, smoking, obesity. Endocrine disrupting chemicals are just one of many environmental variables which we have to consider, and we have to put these, the endocrine disrupting chemicals, in a perspective with everything else we're exposed to. So we have this situation: preconception, fetal, childhood exposure to endocrine disrupting chemicals, and a whole host of other environmental variables such as nutrition, stress, physical or psychological, immunological stress, alcohol, smoking, disruptions of circadian rhythms. All of those have the potential to affect fetal development and childhood development in terms of brain neurogenesis, metabolism, birth weight, and reproductive function. And changing things early on in life can have long-term effects in the adult on brain, behavior, physiology, cardiovascular disease, diabetes, and, where we started off, on fertility and the possibility of cancers. So let's review what we know. Certainly, endocrine disrupting chemicals exist. They work by a variety of mechanisms. They can influence a variety of developmental and physiological systems. They can work over acute and chronic time periods. They can affect epigenetic gene expression and be passed on transgenerationally. And we're all exposed to endocrine disrupting compounds in different amounts, different combinations, and at different times of our life. And if we work down from top to bottom here, we can probably monitor exposure levels. We can certainly determine the molecular changes and molecular sites of action of the endocrine disrupting chemicals. We can certainly investigate and analyse some of the cell level changes which take place, and maybe try to have a preliminary understanding on the tissue and organ changes which occur because of the previous effects molecularly at the cell level. Understanding individual health effects And then transgenerational effects, early life effects, and population health effects in particular get more and more difficult to interpret.